Hi, it's Paul Andersen, and in this video I'm going to talk about the action potential, that characteristic depolarization and repolarization of an excitable cell. I'm going to be talking about neurons, but we could also have action potentials in muscle cells coordinating movement, or in cells of the endocrine system, for example, coordinating the release of insulin. So if we think about a neuron is getting information from its dendrites in cell body. Remember, they're connected to a lot of other neurons, and based on the summation of all of that information in neurotransmitters, it's either going to fire an action potential down the axon or not. And so in this video, we're going to think about three different potentials. The resting potential, that's the resting potential of a neuron at rest. It's not really at rest. We then have a graded potential based on the stimulus that that neuron is going to get. And then we may or may not have an action potential. Now that can lead to synaptic activity on the next neuron. I'm going to go through it in the following order. First, we're going to talk about what a resting potential is and how the sodium-potassium pump and the permeability of different ions in the membrane is contributing to the resting potential. We'll then skip forward to the action potential and the importance of voltage-gated channels. And then we'll come back and talk about how chemically gated channels contribute to either the firing of an action potential or not. And so what is potential? Potential in physics is simply a separation of charge. And so if we think of a battery, a 9-volt battery having potential, that means that there's a separation of the charge within the battery itself. And it also means that we have potential to do work. And so if we're thinking about a cross-section of a neuron, it also has resting potential. What that means is there's a separation of the charges. Now that separation of the charges is going to be across the membrane, just on the surface of the neuron, but we could measure the voltage. It's negative 70 millivolts in a typical human neuron. And so when you're seeing pictures of an action potential like this, on either side of the action potential, we can see that that neuron is at rest. Now, it doesn't mean that it doesn't require energy to do that, and so let's zoom into the surface of that neuron and see what's going on. Now, I've got a little mnemonic that I use with my students to remember the important ions on either side of that membrane. I say the neuron is like a salty banana. Most people remember that a banana has a high amount of potassium on the inside. Doesn't mean that it doesn't have potassium on the outside. There's just a higher percent of potassium on the inside. There are also going to be negatively charged anions. For example, proteins on the inside of the neuron are going to have a negative charge. And then since it's a salty banana, on the outside we're going to have salt. We're going to have sodium and chloride ions on the outside as well. They're going to be at a higher percentage. We still have them on the inside, but a higher percentage on the outside. Now, why do we have more sodium on the outside, more potassium on the inside? I'll show you in a little bit how the sodium-potassium pump contributes to that. But as you look at this right now, there's still no potential. We don't see a separation in charges. And to really figure out how that works, let's remove a lot of it and just focus on the potassium itself, because it's really the potassium and the permeability of that potassium that's establishing much of that membrane potential. And so we've got a channel on the membrane surface. It's called a leak channel, and it's a leak channel that allows potassium to move out. And so if we think about where is the potassium going to go, there's a chemical gradient from the inside to the outside. In other words, there's a higher concentration of potassium on the inside than on the outside. And since some of it can leak through, it's going to go in that direction. But watch what happens as the potassium moves out. We all of a sudden see a separation in charge. Since this is a positive charge here, it's moving out. We have a separation of that charge, and it's a relatively negative charge on the inside. We also see the arrival of a new gradient. That's the electrical gradient. Since we've got all these positive charges here, they're going to repel each other. They're going to want to move towards the negative charge. And so as more potassium leaves, you can see that electric charge is increasing. And so eventually what could happen is it could start to flow back in. And so we're going to have movement of potassium inside and outside, but we've got an electrochemical gradient outside. Now, why doesn't all the potassium just flow out? It's because these are leak channels. They don't allow much of that potassium to flow out. Now, if we look at a PHET simulation, and I'll put a link to that in the description down below, they've simulated the passing of potassium in and outside of the cell. You can see that we also have another leak channel right here. It's not as busy, but that is a sodium leak channel as well. And so we have an electrochemical gradient of sodium. Remember, there's more sodium on the outside that's coming in. And so we've got these two gradients that are set up. You can see the chloride's not moving. You can see that the proteins are not moving as well, and that's because we don't have a channel for them. Now, if we were to just let this go, eventually we would lose that potential because all the sodium would come in. And so we have a sodium-potassium pump where we cash in a little bit of ATP, and that's going to move that sodium out, 3 sodium ions for every 2 potassium ions. And so as this is moving and establishing that membrane potential, again, it's the permeability of these two ions that's establishing it. That sodium-potassium pump is essentially keeping up with the sodium that's flowing in and the potassium that's flowing out. And so that is our resting potential. Negative 70 millivolts is established because we have a separation of those ions on the inside and the outside, and their different electrochemical gradients are establishing that difference in the charge, and so we have this resting potential. Now, it requires energy to do that, but what could that resting potential eventually lead to? An action potential. Now, let's look at what an action potential looks like. This is that PHET simulation again. And you can see there's a huge amount of motion between the ions on that surface. And as we zoom in, we see our sodium leak channel, we see our potassium leak channel, but we see another couple of channels that are really, really important. And those are the voltage-gated channels. How do they work? Well, let's look at the sodium voltage-gated channel. So it looks like this. It's got a little polypeptide ball on the bottom. And so when the voltage approaches negative 55 millivolts, it will open up. And it opens up wide, and that allows a huge amount of sodium to come into the cell. You can see that rush of sodium in that simulation. Eventually, as we move away from that voltage, it's going to be inactivated, and then it's eventually going to close. There's also potassium voltage-gated channels as well, and they're going to open when the voltage becomes around 30 millivolts. They'll open wide open. What is that going to do? It only allows the passage of potassium. Potassium is going to move outside of the cell. And so as we look at that action potential, once we hit that threshold at negative 55, all of those sodium ion channels, the voltage-gated channels, are going to open wide open. What does that do? It allows a rush of sodium into the cell. What does that do to the voltage? It's going to depolarize that neuron. The voltage is going to increase radically because we have an influx of all of this sodium ions. What eventually happens is we get to 30 millivolts, and those sodium voltage-gated channels are going to become inactivated, and now the potassium channels are going to open wide open. What's going to happen? We have a rush of that potassium outside of the neuron. It takes a while for those to eventually close, and so we then have an undershoot. And what's happened is at this point, the voltage has gotten really, really low, but now that permeability, again, those leak channels are going to reestablish that gradient again to negative 70 millivolts, so we could have another action potential. And so if we look at what that looks like in slow motion, we've got a quick opening of the sodium voltage-gated channels. You can see an influx of sodium from there and adjacent. That's followed quickly by an opening of the potassium channels. We then have that outflow of that potassium, and then you can see that permeability is going to reestablish us at that resting potential. We could look at what this looks like as we graph the potential over time. Open of sodium, we see a rapid depolarization, opening of the potassium, a repolarization, and then we have that undershoot. And during that time, until we reestablish that gradient at resting potential, we can't have another action potential. Now, so far, we've only been looking at one cross-section of the neuron itself. How does that message move all the way down the axon? Well, if we look at a simulation, it looks like this. Let me slow that down. What really is going on is we're having depolarization of a segment of the axon, which triggers depolarization of the next segment, and the next segment, and the next segment. So let's zoom in and look at those voltage-gated channels and see what's going on. So if we look at, this is near the axon hillock at the beginning of the axon itself, we've got some voltage-gated sodium and potassium channels. And so when we hit that threshold, those sodium channels are going to open wide open. We have an influx of sodium ions here, and what they're going to do is they're going to move away from each other since they are going to repel like charges, and they're going to change the voltage in this next segment. What does that do? It hits threshold again and opens up the next sodium channels. Now, why isn't it going to move to the left? Because to the left, we have that repolarization of that segment to the left. So we're opening up those potassium channels, and we have an outflow of those charges. We're not going to be near that threshold for the sodium voltage-gated channels. And so what we're really having on the leading edge of this is a positive feedback loop. We have more sodium coming in, which triggers more opening of those sodium channels. And behind that, we have the undershoot of the as we reestablish that gradient again. And so it's going to travel all the way down the axon. Now this is a simulation of what that electrochemical signal looks like as it moves down. But we have some really long axons in our body. We have an axon that goes all the way from the base of our spinal cord down to our foot. And so it's not going to travel fast enough. And so in a lot of those neurons, they're myelinated. And what that means is they're wrapped with myelin, which insulates that nerve fiber. And so if we look at it like this, what's really going on is we're putting those voltage-gated channels in between that myelin sheath. And I'm not going to get into the specifics of what's going on in here, but if we look at this simulation right here, what it does is it allows that electrochemical gradient. You can compare the two to travel more readily as it moves farther down the neuron. Now, action potentials succumb to what's called the all or none law. And what that means is that an action potential will occur or will not, and it doesn't depend on how much of the stimulus it gets. It depends on if you hit that threshold or not. And so once we hit that threshold, we can move towards it, we can move away from that voltage. But once we hit negative 55 millivolts, we're going to have an action potential, and that action potential is going to look exactly the same. Now, in this diagram right here, we have the three different potentials that I'm talking about in this video. At the beginning, we have the resting potential. Remember, that's contributed to by the permeability of the membrane to different ions. We then have the action potential, that rapid depolarization and repolarization of the neuron. But what's going on in here, we have graded potentials. In other words, the potential is moving towards or away from that threshold. And so if we think about what's going on there, to really understand that, we're getting information from other nerves. There are synapses here, and those synapses are releasing neurotransmitters. Those neurotransmitters are moving across the synapse, and they're opening up a new form of channel that's important when we talk about graded potentials. Those are chemically graded channels. And so these neurotransmitters are going to dock with proteins on the postsynaptic side of those neurons, and what they'll do is they'll open it up. What does that do? Well, it depends on what type of neurotransmitter it is and what type of receptor it is. There are some that are going to be excitatory. And if we look back at our graded potentials, excitatory means that they're moving us towards the threshold, towards that all or none action potential to occur. What's the easiest way to do that? If we open up the channel and we have a channel that allows sodium to run in, that's going to depolarize us and it's going to move us to that threshold, just like those sodium-gated channels are going to do. Now, we also have inhibitory neurotransmitters and receptors. What are they going to do? They're going to move away from that threshold. What's the easiest way to do that is to have a receptor that's going to allow potassium to rush out, or you could allow chloride to come into that neuron itself, and it's going to push us away from that threshold. So we can think of a neuron as an analog portion and a digital portion. So since it's all or none down here, where you're either going to have the action potential or not, so it's like a 1 or a 0. But if we look on this side of the neuron where the cell body is and the dendrites are, we're getting a lot of information from neurotransmitters of neurons that are attached to the dendrites and the cell body of this neuron itself. Some of those are excitatory. What they're doing is depolarizing that neuron. They're increasing the voltage, pushing us closer to that threshold. And some of those are going to be inhibitory. They're pushing us away from it. And so it's the summation of all of those signals that's telling that neuron to either fire an action potential if we hit negative 55 or not. And so that is the all or none law. And so in summary, what is the resting potential? It's negative 70 millivolts. It's where a neuron is sitting. It has a potential to do work. It's established by the permeability of the membranes and the sodium-potassium pump. We then moved on to the graded potential, and that's based on all the neurotransmitters that we're getting from connected neurons. And then if we hit that negative 55, we're going to have an opening of those sodium ion channels, and we're going to have an all or none action potential that runs down to the neuron. So that's the action potential, and I hope that was helpful.